So again, without going into the details, I'm going to go over the FASTA algorithm quickly. FASTA, similar to BLAST, it starts looking for certain sub-windows of exact matches. They are called K-tuples or K-tuples. Words of K is used, so words of length 1 or 2 for proteins and 4 to 6 for DNA sequences. So it does, so it finds these exact matches within the two sequences, and it doesn't do an extension similar to BLAST. So it selects these exact matches. If we do it in the, if you look at like the dynamic programming matrix, okay, if one sequence is here, the other sequence is here, these exact matches, they are going to appear as diagonals, as local diagonals in the in this matrix, right? So such a match means that this region of this sequence matches exactly. So this region here matches exactly to this region here. This diagonal indicates that. So it's after it finds these matches, it finds the top 10 scoring best matching local diagonals with matches and mismatches, but no gaps. So it does this extension locally for these both of these sequences after finding these K-tuples, and then it uses kind of a dynamic programming approach around the top 10 scoring regions. So the efficiency, the speed up over the original dynamic programming algorithm is achieved by not filling in the entire table. It fills in the table by just looking at a certain narrow band around these local matching regions. So that's the main thing about FASTA you should know. And while finding the exact matches, we may do something like this. We may, so in this example, we have one tupes, okay? Okay, meaning that we are looking for words of length 1 in the amino acids. So this word, amino acid A, this word of length 1, appears in this position, okay, in this protein sequence, and in this position in the other protein sequence. Okay, so here the position, the first position here is, okay, so actually this should be starting from, so this 1, 2, 3, 4 should be indented towards right 1. So there's a typo here. This N is the first amino acid, C is the second amino acid, and A is the sixth amino acid. So its position, we store the positions of these words in both proteins, and then if we have a matching region, for example, we can see it as offsets that are going to be agreeing with each other. So it's like this, after we record the position of the occurrence of each word in both protein sequences, we can find out the deviation in this position between the protein sequences. This is 0, they both occur in position 6. But if you look at the C, okay, what we hear is we see that this C, SP, occurs exactly in the other protein sequence, but in a different position. This CSP starts at the second amino acid in protein A, but it starts at 7th amino acid in protein B. So the offset, the difference between these two starting positions is -5. And we see that this -5 occurs between the occurrence of these words in a number of times. So we can, by collecting these together, we can quickly find that by finding out the common offset words, we can find this matching region CSP between the two proteins. And after that, after finding all such matches, we may have multiple common offsets. Okay, this -5 is not, may not be single. We may have other matching which suggests -2, -8. So what we do is we find these top scoring diagonals, find the 10 best diagonal runs which have the longest matching regions, and we rescan them and we score them with the scoring matrix like Blossom or PAM. FASTA uses PAM scoring matrix, and we cut off the end points which have mismatches, okay, to achieve the highest scores, and we join also regions that are consistent with gapped alignments. This is done after finding. This is done by looking at neighboring local diagonals. If we can put a small number of gaps in between them that can be joined, like this, for example. Look at here. These two regions can be joined if we have a gap here, a small gap here. Similarly, these two regions can also be joined. After doing this joining these regions that are consistent with gapped alignments, we run the dynamic programming global alignment centered on the best initial region. Okay, so after finding the best initial region, the matching sequences, we run the dynamic programming only in a narrow band around the initial region. So that's the FASTA also runs the dynamic programming algorithm, but it gains certain efficiency, speed up, by not filling in the entire table, by just looking at, by filling in the certain entries that are, so it's like FASTA first tries to get an intuition of where is this, in this whole table, our alignment, the best alignment between these two sequences is going to be a path in this table, right? So FASTA gets an idea about where this path is. So it gets an initial idea about the existence of the location of the path, and if you find the certain heuristic about the location of this path, you don't need to fill these parts of the table. You can only focus your attention onto this region, and this is what FASTA does to gain a certain speed up. And this speed up, discarding these regions, basically reduces the ON square complexity to almost a linear complexity. Okay, the path length is on the order of n, or n plus m at most. So this tries to convert the quadratic running time, reduce the quadratic running time of dynamic programming to linear time dynamic programming. So this is how it achieves the speed up. Okay, so it doesn't use this, for comparison, it doesn't use the statistical basis that much compared to BLAST. And again, it begins with exact matches. The local diagonals are exact matches. And its quality and complexity usually similar to BLAST. Something that FASTA provides more higher quality alignments because it uses dynamic programming to do the alignment. And something that BLAST is faster than FASTA, unlike the name. Okay, so the FASTA says fast in its name, but BLAST may be faster than FASTA. Okay, and here's the history of sequence searching at the end. In the 1970s, the first global alignment of sequences. It took 10 years to add 0 to the recurrence relation. The local sequence alignment, the Smith-Waterman case. It was a very simple modification to the first recurrence relation, but such simple things may not be very easy to find. So Smith-Waterman proposed this addition to the modification for the local sequence alignment. 1985, FASTA was proposed, and 1989, 1990s, BLAST, original BLAST, and BLAST 2 is 1997. Okay, so that's it for sequence alignment and sequence searching. Do you have any questions before we continue to statistical significance of alignments? Okay, so let's move on. We are like actually more than 1 week behind schedule, but don't worry about it. We are going to catch up. Okay, so this one has the faster. Yeah. Now, as a reminder, do not forget that the purpose of sequence alignment is to look for homology. Whether a given protein sequence or DNA sequence is homolog, they share the same ancestors, whether they share the same function, and this is what we're looking for, okay? And this is sequence alignment is a very important tool for this purpose, for function identification. If we can identify homology, we can infer function. If a homolog protein in a well-studied model organism has a known function, probably we can transfer this information to the new organism as well. And in 1997, this archaeobacteria, it's difficult to pronounce it, Jannaski, or I don't know how to pronounce it, but it was a newly sequenced organism, okay? And about 70% of its genes, although it is a newly sequenced organism, researchers were able to identify the functions of 70% of the genes based on sequence similarity only. So these genes had similarities to other genes with known functions on other well-studied organisms like yeast. So the model organism, the word model organism is used, so there are certain species which are easy to study with, okay, in the lab. These are called our model organisms to conduct studies on other areas, less common organisms. We usually conduct research on biologists. Usually conduct research on these model organisms because they are easy to study with, and the most commonly studied model organism is baker's yeast. Or Saccharomyces cerevisiae. This is the Latin name for baker's yeast. It is many researchers, maybe thousands of researchers have studied on this organism. We know a lot about this organism, about the functions of its genes. And this yeast is an eukaryote, right? But it's a single-cellular organism, single-cellular eukaryote. So I'm looking at biologists to, because I'm not sure whether my biology background is not that good. So, in addition to this model organism, there are multicellular eukaryotes, which are also very common, like the C. elegans organism. Its common name is nematode worm. It's a very small worm, millimetric dimension. I mean, it's very small, but it's multicellular, and it has common genes with humans as well. And the other model organism, Drosophila melanogaster, which is the common name is fruit fly, is also, so these are, fruit fly is also for developmental stages, gene regulation is studied on Drosophila a lot. And there are model organisms in plants as well. And there are also studies, direct studies on humans. So human genes are also, there are certain cell lines. For example, there is a certain, there are human cells that are cultured, like I know there is this HeLa cell lines, H-E-L-A. It's from a cancer patient, okay? And they've been growing these cancer cells on test tubes. Maybe it's more than 20, 30 years. It's very old. Okay, these cell lines from a cancer patient taking 20, 30 years ago, they've been growing these cell lines, and it's copied, it's duplicated all around the world. And many people are researchers. You can buy these cell lines. There are different cell lines as well. So you can conduct studies on human cell cultures as well. Okay, so studies, many studies on these model organisms help us to find the functions of their genes. And if we have a new organism, which is not a model organism, we can infer the functions of these genes based on its similarities. If we have a homolog gene in one of these model organisms, it will help us to infer the function of this new organism. Now, but the most important question in inferring such function is that how much sequence similarity do we need to state that to infer that these sequences are homologs? They have the same function. So how much sequence similarity is enough? 50% sequence similarity, 60%, 30%? And the answer to this question is there's no unique answer. There's no universally accepted answer, because there are sequences, there are protein families known to share common structure and function with only 10% sequence similarity. But this does not mean that when you have two sequences with 10% sequence similarity, it doesn't mean that they are going to have the same function. They may be completely from different families. Okay? I mean, there are examples in which 10% sequence similarity, they are homologs. They are known to be homologs because we know their structures. Their structures are very similar, so they must be homologs. But when we look at their sequences, only 10% similarity. Okay? But this does not mean that when you find two other sequences with 10% similarity, they may be completely unrelated. Okay? So it tells you that this is a very difficult problem. We cannot have a single measure that tells us that a certain homology threshold level. Usually the sequence similarity of between 20% to 30% is called the twilight zone. We can say whether it's, we cannot directly say they're homologs, but they can be. But usually if you have a sequence similarity more than 50% sequence similarity, they're most likely to be homologs. So for sequences that have very highly similar regions, but you also saw, okay, I said 50% similarity is usually a good, and they should be functionally homologs. But remember our made-up query? It contains 62% similarity to a database protein. So it means that just a random sequence may have 62% similarity to a known protein. And of course, it doesn't mean that the protein we just made up is going to have that function. No such protein exists, okay? So, but the percent identity, the 62% similarity, it doesn't tell us much. The e-value in that query, which was 0.29, was much more meaningful than the percent identity. Okay? If you were just looking the percent identity, you would say that, oh, 62% similarity. We would say, oh, actually, that 62% was not the similarity. It was the 62% of the region of our protein sequence was aligned to the other one. Probably the percent identity was much lower, but even if it didn't tell us much, but the e-value told us a lot. It told us that there's a high chance that you got this score by just random, by just chance. Okay, there's a high probability that you got this match by chance. Now, and our goal is to find out whether is this really a homology or is this similarity by chance. So this is the two things that we need to distinguish from when we find a hit to our query. Is it similarity by chance, or is it true that they evolved from a common ancestor, hence they have similar function. Are they real homologs, or what we have here is just a random hit? Okay, so this is the most important question biologists are trying to find when they're doing sequence searches. And there are recent studies in the last 5-10 years. There are studies that shows that sequence similarity itself may not be a tool accurate enough to infer homology. We may need some additional tools like phylogenetic analysis. For example, imagine we have a set of hits. Okay? Imagine we perform a blast query. We have 100 hits to our query protein. The first one says this protein is, for example, a certain function, function A. The first hit says that it's a protein with function A. And then the second, third, and fourth hit says that this protein is a function which has function B. Okay? Now, what are we going to infer as function? Is it function A or function B? So, many people use some additional tools like what happens if we, for example, analyze these first 100 hits with respect to each other as well. If you look at these first 100 hits, are there certain functional groups which are apparent, and maybe a certain group itself may be much closer to our protein, even if a single member of the other group is the closest. For example, if you look at these function A and function B, if we have performed a more detailed analysis on the results, we could have maybe seen that many function B proteins are on average distance to our query protein, but the function A on the average is distant from our query protein, but there may be a single member which is the closest. So it may be wiser to infer the function of this protein to be function B, although the best hit has function A. So, and maybe also function A and function B. These two functions may also be related because they also, both of them came up as hits. There may be relationships between them. The reason I'm telling you this is that the evolution is really complex. Okay, these proteins functions, they may be related. Proteins share domains with each other. Domains are split up into two different proteins. There are multi-domain proteins. So it is a very complex thing. You can, by just looking at sequence similarity and by just looking at a single number like e-value, you will get some idea about function, but it doesn't tell you the certain homology. You need to do some detailed biological investigations analysis to be more certain about your function, the inferred function. Okay, so this is just as a precaution. We do not use sequence similarity alone these days to infer function, but it gives us a lot of clues. And today, in the last 20 minutes, I'm going to talk about statistical significance here. And the reason is that the measures of similarity, the score values we use or the percent identity, does not give us good information about homology. For example, with percent identity, given two sequences, there may be 40% similar, 70% similar. What problems can you think of in terms of inferring homology with percent identity? The first example I gave you is like 10% identity may actually be there are homologs with 10% identity. That's one problem. Can you think of other problems with percent identity measure? Meaning you have two sequences, how much identical they are. This is what percent identity means. Is it a good measure, or can you think of some problems about this measure? Any ideas? Yeah, but most of the other parts may be different, right? So so percent identity oversees local similarities. So this is, if we have a local, for example, they may both be multi-domain proteins, but they may share a certain domain which is important for its function. And the percent identity, it's a global measure, so it cannot capture local high similarities. And scoring matrices also, the problems with scoring matrices is that these values are built. The score of I to V, for example, being 10, that score is found out by looking at some example alignments on a certain protein family. So it makes certain assumptions about amino acid substitution rates. It looks at, oh, this I amino acid has been changed to L amino acid this much, so let's give it a high score. But in another family, this amino acid substitution may not be observed at all, which means that it may not be a good idea to give a high score to that two amino acid mismatches in that protein family. So therefore, using universal scoring matrices have these problems as well. And another problem is, the scoring matrix, it gives you a score. Okay, score of 150. Is it a good score or not? 150, what does it mean? So what is the norm here? So the percent identity, it's between 0 and 100. It's better than the scoring matrix in that regard. I mean, we have a universal scale. Okay, 100% is the best. 0% is the worst. But what about the score we get from an alignment implied by a scoring matrix? Score of 200, is it a good score? 300? So we cannot really have an idea of scale, especially if you have long sequences. Long sequences tend to get a higher score compared to short sequences because these scoring matrices, they're additive. Okay? You add the scores up, you can get very high scores if you are adding scores. So longer sequences are going to get higher scores compared to shorter sequences. So you cannot have a universal global scale using scoring matrices. So to overcome these difficulties, one thing we can use is statistical significance. Statistical significance, it's not only used in sequence similarity, we use statistical significance for many different things. And unfortunately, as computer scientists, we usually do not use statistical significance that much. When we report our results, we just report them, okay, these are my results, this is what I found. And we do not usually compute significance, which is, I think, a bad thing. Because significance tells us, the main thing that tells us that, okay, your algorithm may have produced some result, okay? Imagine you have there's a certain optimization problem, okay? You want to maximize for example, the value of items that you put in a bag, okay? You devise an algorithm for that. And your algorithm spits out some results, but you have no way of finding whether the results of your algorithm is good. I mean, there's no way. Can there be another algorithm which produces better results? You don't know that, okay? So this exhaustive search is not possible. So you have a certain problem, you write a program, your program produces results. So would you just accept those results are as good? So can you do something better? So the problem here is that you have an algorithm which gives you some results, but you have no idea about how good these results are. Okay, you filled in something in this bag with certain values, but can another algorithm do better? So this is a very important question, and by measuring the statistical significance, we give something like this. If I had a random algorithm which just puts the items randomly in a bag, okay? This is our baseline algorithm. I mean, without no intelligence at all, just put random items in a bag, and this is the cost, this is the profit I get from putting these items in the bag. Now, if your algorithm is intelligent, it should produce something better than this cost, right? I mean, otherwise, why spend your time thinking about this intelligent algorithm? You could have just used the random one, which gives you some results. So You can think of statistical significance like this. It's a measure which tells you whether the result produced by your algorithm, how unlikely this result is from a random algorithm's result. It is going to, therefore, you will be able to tell that, okay, a random algorithm produces this cause. The profit I get from my algorithm is very, very unlikely to come from a random algorithm. So this kind of a measure is comforting, right? It means that you did something intelligent in your algorithm. You could not get it by just random search. It's very unlikely to get this from the random. So this is what statistical significance provides. It provides us whether the thing we found is something really valuable or we just got it by chance. And there are different measures of statistical significance, okay? And usually for sequence similarity, we want to ask this question. How different is our result from a random match, okay? Or a match between unrelated, non-homologous sequences, okay? So our sequence aligned or our blast search gives us a hit. Okay, here's a hit between the query and a database sequence. And what we wonder is, how different is this result if from two unrelated sequences, from completely unrelated, non-homologous sequences? And we can also ask the question in a different way. These are similar questions, but it's a different question. So given a set of sequences not related to the query, so this is emphasized, okay? So if I know a set of sequences not related, for sure, if I know these sequences are non-homologous, in this set of sequences, or a set of random sequences would also be not related to the query, what is the probability of finding a match with the same alignment score produced by your algorithm by chance? Okay, this probability, so this is different than e-value. This probability is called p-value. It tells us it's the probability between 0 and 1. If that probability is, for example, 0.7, it's very bad. With just 0.7 probability, you could have gotten a same score or better score from a set of completely unrelated sequences. So how can you trust your score? You cannot trust your score because you could have get the same or better score. It's 70% chance. I mean, if you throw a coin, you could have got an unrelated sequence. So your algorithm itself, the result itself, may be completely unrelated. So these are the three main statistical measures that are used. Okay, the p-value, e-value, and z-score. So p-value is exactly defined as the second question. It gives you a probability and the probability to get at least the probability that there is a sequence, a single sequence that can achieve the same or better score by chance. Let me give you an example of the p-value. For example, we can let's get back to the previous problem. So we have a bag, okay, and we have a set of items, okay, and we fill our bag with this set of items. And we want to find out the p-value of result provided by an algorithm. So we have an algorithm, and this algorithm takes as input the set of items, okay, the set of items, and the bag size is taken, so the bag is taken as input, and it produces, it says that you need to put these items in the back. Okay, this is the input of your algorithm. This is the output of your algorithm. Okay, and it also tells you these are the items you put in your bag, and it gives a numerical value, profit. Okay, the value, or let me say value. So the value of the total value of the items. So the total value of the items in the bag is also produced as output. Now, this is your algorithm. Now, if you want to compute the p-value of this value, what are we going to ask? The question we ask is this. The result produced by this algorithm, what is the probability that a completely random placement of items in the bag will produce a better or a same or better value? Okay, this is the exact definition of p-value here. What is the probability that we could get same or better value by chance, by just putting these items randomly in this bag? Of course, we have certain constraints, for example, the bag size, but just start until you fill in the bag, just fill in these items until you fill in the capacity of the bag, for example, randomly, without using any heuristics or algorithm, just put randomly. And what is the probability that this random filling of the bag can produce a value better than this? And if our algorithm is really a good algorithm, we expect this p-value to be small, right? I mean, the smaller it is, the smaller the probability is, the better your algorithm, the more different your algorithm is from random. If your algorithm produces similar results to a random algorithm, then what's the use of your algorithm? Just fill in the bag with random values. So here's how we can compute the p-value. Okay, this is, and I'm going to stop after this. What we can do is, by providing different test cases, okay, of maybe the same size or on the same test case, okay, on the same set of items on the same bag, we run the random algorithm a number of times, okay, like 1,000 times. We run a random algorithm, so this is the random one. We give input to this random method to fill in the bag, okay, and spit out some values. And we do this 1,000 times, so we get some random values. So the main reason of doing this is to get an idea about what random outputs look like, okay? To get an idea about the distribution of a random output. So it's, if you run this problem set, if you fill your bag randomly, this is what random gets. We need to get an idea about this. Why do we want to get an idea about this? Because we want to find out how different is our algorithm's result from random algorithm's result. Okay, so in order to be able to find this difference, we need to have an idea about how does random look like. Okay, if the random algorithm produces always scores 0, 0, 0, 0, 0, then it means that if you find something other than 0, it means you are different. Okay, so in order to get an idea of how does random results look like, we need to generate some random results and look at the distribution. So this is what we do. We run this random algorithm, for example, 1,000 times or 10,000 times, and at the end, what we are going to get is we plot the results in a histogram. So the x-axis is our value or profit, okay, increasing from left to right, and the y-axis is the count of random runs that produce this exact. So for example, what we have here is, for example, if this is the profit, the value 5, for example, this many 10 runs produced a value of 5. Okay, this is a histogram. And usually what we would expect is we would get a graph like this. Probably something like this. Okay, I'm just guessing. Okay, this may be completely different. But imagine we have something like this. What does this histogram tell us? It tells us that, and this is the value, for example, 20 here. Okay? Sorry. Actually, I did this in the wrong way, I guess. So we should have the... No, it's correct. Yeah. Okay, what does this tell us? Okay, this tells us that there are many, okay? There are 20 of these random runs. So there are a high number of runs which produced very low profit, okay? There are many random runs which produce low values, and there are some, for example, one of them produced a high value with 25, okay? Sometimes random values could also produce high profits, but most of them produce low profits, and there are, due to this randomness, by chance, sometimes you can get a high value like 25. Okay? Now, after we have such a histogram, let me just increase this to here, like this. We may fit a graph or we may hit a polynomial function on this histogram. Now, what I'm going to do is, after we have this histogram, imagine our algorithm produced a value of V. And this V fits here. Okay, this is our algorithm's output. Now, if I normalize the area under this curve to be 1, what I have is a probability distribution. Okay, if the area under this histogram is 1, it's like I'm going to have what is the probability of obtaining a certain score by a random algorithm can be modeled by this histogram. And the p-value is going to be exactly, if the value produced by my algorithm for this input is V, the area to the right of this curve is going to be my p-value, because it's going to be the probability that a random algorithm can obtain a better or same score than my algorithm. Because my algorithm scored V, and there are this many random runs that could produce better results than your algorithm. And the smaller this area is, the more different our result from the random population. So it's like we have a certain, now we have an idea of how random looks like, and we put our results here, and we say, okay, our result is much different. It's much better than most of the random results. Only a very small number of random results can actually exceed us. And the smaller this area is, the better our algorithm is. Okay, this is the main, this is how you can compute p-value by first modeling your random distribution and finding out putting your actual result here and compute the area under the rest of the curve to see what is the probability that you could have. I mean, if this probability could also as well be 0. I mean, the area to the right of the curve, 0 means that if this V, for example, was somewhere here, where after the curve meets 0, maybe, if your V is here, no random algorithm can obtain a better score than you. The p-value could be 0, which would mean a very good thing. Okay, so this is p-value. E-value is not a probability. It computes the number of random runs that could have produced a better value. And z-score is this. Z-score is similar to the way we use the curve analysis to give you your grades. Z-score just tells how much standard deviations this V is away from the average of this random distribution. If the average is 50, for example, and the standard deviation is 10, if we get a score of 80, it means that we are 3 standard deviations higher than the mean of this. This is going to be our z-score. Okay? Z-score measures how much standard deviation away you are from the mean. And it can be a negative number. You may be lower than the mean. So minus, you may have minus z-scores. And e-value is the count how many of these random values could be higher than you. And p-value is the probability. Okay? What is the probability that a random number, a random run can exceed the scoring value? So that's it for today. We are going to continue how we can compute these e-values for sequence matches. So sequence similarities, how can we compute an e-value for them? We are going to continue next week about this.